Hello everyone and welcome to today's live broadcast, Circulating Biomarkers, What Matters for Isolation of Biomarkers from Liquid Biopsy Samples, presented by Dr. Verena Schramm, a Global Product Manager at QIAGEN. I'm Susie Valdez of LabRoots and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. QIAGEN serves more than 500,000 customers around the globe, all seeking insights from the building blocks of life, DNA, RNA, and proteins. They deliver samples to insight solutions for molecular testing, propelling QIAGEN customers from start to finish to unlock new insights. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located on the lower left of your presentation window and type those questions into the box that appear on the screen. We'll answer as many as we can during the presentation. Also, notice that you're viewing the presentation in a slide window. To enlarge that window, click on the screen icon located on the lower right. If you have any trouble hearing or seeing this presentation, click on the support button at the top right or use that Q&A button and let us know you're having a problem. This presentation is educational and offers continuing education credits. Please click on the button in the lower left to follow and process those credits. Without further ado, please join me in welcoming Dr. Verena Schramm. I will now turn the presentation over to our speaker. Welcome, Dr. Schramm.
Thank you so much for the very nice introduction, Susie, and thanks to everyone for joining this session today. I want to talk a little bit about circulating biomarkers and what matters for the isolation of these biomarkers from liquid biopsy samples. QIAGEN products that I will be talking about today, shown here, are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. This is the agenda for today, and I really want to set the stage by giving you a short introduction to liquid biopsy and remind everyone of what this technique is about. Here is a common definition of liquid biopsy: the application of minimally invasive techniques that can be used to help detect molecular biomarkers in body fluids, which may allow for improved diagnosis and treatment of cancer as well as other diseases. So this definition already tells you that liquid biopsy is a minimally invasive technique and holds really a great potential for diagnosis and treatment of cancer patients and other patients in the future. The advantages of liquid biopsy in comparison to the classical tissue biopsy are listed here on this slide. First of all, liquid biopsy has a lot of advantages when it comes to sample availability. It's much easier to take a blood draw from a patient or to take a urine sample from a patient than to obtain a tissue biopsy. Also, the process simplicity is advantageous for liquid biopsy compared to the tissue biopsy. The processing speed of the sample also is better for liquid biopsy. With liquid biopsy, you can really early detect or even screen for diseases like cancer, whereas with a tissue biopsy, usually the diagnosis of the patient has happened before, and afterwards a tissue biopsy is taken to really diagnose and characterize the cancer. Disease monitoring is another advantage of liquid biopsy. Because you have so easy access to liquid biopsy samples, you can constantly monitor the disease progression. That is especially important again for cancer patients. Liquid biopsy also has an advantage when we talk about cost effectiveness. So taking a blood draw or taking a urine sample or another body fluid sample is a lot easier than taking a tissue biopsy, which usually requires a small surgery. In terms of the sensitivity, there is still a little bit of discussion in the field, and the sensitivity of these technologies clearly depend also on the analytical methods that are used. However, now, especially with methods like next-generation sequencing or digital PCR, the sensitivity is really high enough to detect all sorts of mutations from a liquid biopsy sample. So there is more and more research being done on liquid biopsy, and people move away from the classical tissue biopsy, so to say. There is a lot of potential also to then look into circulating biomarkers. So circulating biomarkers are the biomarker molecules that you can obtain from a liquid biopsy sample. First of all, circulating biomarkers is a field that is really well funded. Especially exosome research is one of the best funded research areas at the moment. And there is a lot of publications coming out focusing on circulating biomarkers, and people are finding new biomarkers essentially every week. This is also highlighted here by the lower right graph, which shows you the increase of publications from PubMed on the topic of liquid biopsy and circulating biomarkers. And as I already mentioned, we now already have the right toolbox in hand with sensitive assays like next-generation sequencing or digital PCR to really analyze these liquid biopsy samples and the circulating biomarkers within these samples in detail and understand the full potential of this new sample type. However, there are also a lot of challenges associated with the analysis of circulating biomarkers. Usually, these types of analytes are only present in very low sample concentrations. That is especially true for circulating RNA obtained from a liquid biopsy. There is also still a lot of analytical errors that can be caused by sample handling, and this is linked to the fact that there are no SOPs or standard working procedures in place already, just because liquid biopsy is such a new area and people are still actively working on defining such standards. Excuse me. When we look at liquid biopsy, there is really a broad variety of different biomarker types that can be investigated. The types of analytes that are commonly studied are listed here on this slide. Circulating cell-free nucleic acids is probably the type of analyte that's been studied the most in liquid biopsy context in the past years. However, also exosomes as well as circulating tumor cells are becoming more and more important for the analysis of new biomarkers. I want to talk a little bit more about exosomes and also focus on circulating RNA biomarkers in the further of this presentation. So exosomes are small vesicles that are excreted by all cells of the body, including tumor cells. Subsequently, they can be found in all biofluids that can be obtained from a human body. Extracellular vesicles, or exosomes, are cargo molecules, and they play an important role in cell-to-cell communication. They actually are found to contain stable nucleic acids, in fact all sorts of nucleic acids that you can also find in the normal cell, so mRNA, microRNA, other regulatory RNAs, as well as DNA, proteins, lipids. And all of these different analytes are in fact protected from degradation by the lipid bilayer that is, let's say, surrounding the exosome. The interesting point about exosomes and why people study RNA from exosomes is that the RNA contained within exosomes provides a complete snapshot of the transcriptome of the secreting cell. So again, it's a minimally invasive way to look at the expression changes that are happening in a specific cell type that you're interested in. The contents, however, really varies also depending on the physiological or pathological conditions. So similar to other features, if you compare the exosomes from one human to another, they will be very different depending on these conditions. Cell-free RNA is extremely important because extracellular vesicles are also secreted by tumor cells. So the RNA that's contained within these extracellular vesicles, in fact, can be used to detect tumor-specific mutations. These tumor-specific mutations, like fusion genes, for example, are only detectable at the RNA level. So looking at the DNA level, you will not be able to find such fusion genes. You really need to look into RNA as a nucleic acid type. There is also a lot of microRNA content within these exosomes, and we know that the microRNA content that is within exosomes is different and very specific to that compared outside, to the microRNA content outside of exosomes. And there is a lot of publications that are focusing on RNA in the context of EV research, and more are coming. So although past research has been focusing more on the cell-free DNA side, RNA analysis, especially in exosome research, is becoming more and more popular, and more people are looking into this in more detail. This is just a reminder of what RNA biomarkers are present in liquid biopsy samples and where they are typically found within a liquid biopsy sample. So mRNA is usually only contained inside of extracellular vesicles, because this is where mRNA is in fact protected by the lipid bilayer. Long non-coding RNAs are also exclusively present inside of extracellular vesicles, again because they are protected there. For microRNA, it's a little bit different. MicroRNAs in liquid biopsy samples are either within extracellular vesicles, or they are bound to protein complexes like the ARGO2 proteins. So if they are outside of vesicles, they are protected by protein complexes, or they are protected within exosomes. Let's talk a little bit more about what matters for sample collection and disruption. So this is a classical RNA workflow from sample to insight, going through all the different steps that you have to consider if you want to do a gene expression analysis based on RNA in any sample type, in fact. And I want to remind everyone here that each of these steps is very important, and that a certain amount of detail needs to be paid towards each of these individual steps. Actually, the same applies for liquid biopsy samples that we are focusing on today. So let's talk a little bit more about the sample collection and also sample disruption. I was already mentioning that the RNA that's relevant for liquid biopsy analysis is in fact well protected. So mRNA is encapsulated and protected within extracellular vesicles and exosomes, and extracellular microRNAs are either contained within exosomes or loaded into high-density protein complexes like ARGO2. Again, this makes them very inaccessible for RNases, and they are thus prevented from RNA degradation. The challenges of RNA isolation from liquid biopsy samples are more on the side of low RNA content and also a high inhibitor amount that can be present in serum and plasma samples specifically, which can inhibit the downstream assay. So stabilization of blood cells is in fact more a topic for cell-free DNA than it is for RNA, because the RNA that you're interested in the liquid biopsy context is usually very well protected and preserved. For DNA, the story is a little bit different, because when cells undergo apoptosis, they release gDNA from their nucleus, and this gDNA is diluting the cell-free DNA signal that you would be interested in a liquid biopsy context. So stabilization of blood cells is very important for cell-free DNA analysis. It's less of a concern for RNA analysis from liquid biopsy samples. What is also very important is that you take a detailed look into your pre-analytical workflow. We know that a lot of the differences in results or inconsistencies in results that are obtained very often stem already from the pre-analytical processing of the samples. So here are some considerations for the isolation of intact exosomes and EVs as well as their content. First of all, quite some background can come from cellular nucleic acids that are released when cells undergo apoptosis. So what we usually recommend is to do an upfront filtration step or centrifugation step, as indicated here on the left side in this little pictogram, which removes background from cellular RNA that can be diluting the desired sample. Then another question that we are very often asked is what kind of sample type should you be looking at? Should you, especially when we talk about serum or plasma, people are asking, is it better to use serum or better to use plasma? Based on our experience, we would say that plasma is a better sample type to analyze the nucleic acid content of liquid biopsy samples. But there is also reasons for choosing serum, really dependent also on what sample type might be available for you and for your research. However, it's very important that throughout your experiment or your analysis, you are consistent in the sample type that you're using. So don't mix serum and plasma samples, and avoid this if it's possible. Another aspect to consider is the blood collection device. From our experience, we know that most of the blood collection tubes are suitable for exosome isolation and subsequent RNA isolation and profiling. However, again, here it's important that you ensure to use the same blood collection device or blood tube throughout your experiment and for all your analysis. So let's move into RNA purification topics. Again, here it's important to not just look into RNA isolation, but also QC of the RNA, as this is an integral part of every good RNA experiment, to look into your RNA quality after the isolation. When we talk about RNA isolation from plasma or serum samples, again, the very small amounts of RNA, especially exosomal RNA, are probably the largest challenge for any isolation method. That's in fact why we have dedicated solutions here at QIAGEN specifically designed for low amounts of RNA or specifically for exosomal RNA. In terms of starting volume for microRNA, as it's a little bit higher abundant than mRNA, it's usually sufficient to start with 200 microliters of plasma or serum. You can use higher volumes, however, this also increases your risk of getting inhibitions in your downstream assay then later on. For mRNA, as it's usually only contained within exosomes, the abundances are much lower. So here we recommend to at least start with 1 milliliter of serum or plasma to have then sufficient amounts of RNA molecules, mRNA molecules, to go into a deeper analysis of gene expression differences. Contamination is especially a problem for serum or plasma samples, as especially plasma has a lot of protein contaminants, but also contaminants like anticoagulants, heparin or EDTA can interfere with downstream assays. So here it's very important to efficiently remove such contaminants to avoid inhibition in the downstream assay. Also, another point that I forgot to mention is the presence of RNases. RNases are degrading your RNA and are thus lowering the RNA integrity, which can be an issue to make a significant statement or a good analysis of your sample in your downstream assay. So as soon as you release your RNA in a liquid biopsy sample from its protection, either by lysing exosomes or by lysing the proteins that are binding microRNAs, it's also important to inactivate RNases at the same time. What is usually a very good way to inactivate RNases and also to remove contaminants is a phenylchloroform step. That's why a lot of our kits are still containing a phenylchloroform step. However, we have recently launched a new Munisi Serum Plasma Advanced kit, which uses an alternative to phenyl, which is a combination of buffers RPL and RPP. One is meant for the lysis of proteins and exosomes and the inactivation of RNases, And the second one is then to precipitate the proteins and contaminants and remove them from the sample. In the second step, you are then taking the supernatant and you go into the typical spin column based protocols from QIAGEN. So here you really have the advantage to get rid of the phenol. You don't need to do a phase separation step, and in fact you don't need to work under the hood anymore. These kits are also including what we call the UCP columns, and I'm going to talk a little bit more about that in a second and what exactly it stands for. In terms of the performance, we are here comparing now this new kit and its performance to our legacy Myanese serum plasma kit. And we see that despite getting rid of the phenol step and replacing this step, we are not making any compromises on microRNA yields, as you can see here by the CT values on the y-axis, here shown for 3 different donors. So you are getting an easier protocol and you don't have to compromise on microRNA yields or quality. This new kit is also able to efficiently lyse exosomes, as you can see by this graph here. We are looking at a comparison between the new kit and our ExoRNase serum plasma kit. ExoRNase kits are specifically isolating total RNA from exosomes. So for those microRNAs that are known to be primarily inside of exosomes, we see very similar yields between the 2 kits. For those microRNAs that are present outside of exosomes, we see a slightly better performance for the Myanese kit compared to the ExoRNase kit, and that's because ExoRNase is enriching for exosomal RNAs specifically, and is getting rid of the microRNAs that are outside of exosomes. Let me tell you a little bit more about the UCP column concept. So UCP stands for ultra clean production, and especially in the days of next generation sequencing, people are looking in an unbiased way at the complete sample's nucleic acid content. And with these analyses, it actually became obvious that contaminations, especially in the microbiome context, are coming off from spin columns. That's a fact of the spin column membranes that a lot of providers are using. However, this is then also reducing the number of useful reads that you get from your NGS experiments. And especially for a microbiome experiment, you can even get a certain bias in your result due to this contamination. So what now a lot of our columns are undergoing is the UCP treatment. So that is a special production treatment which ensures ultra clean alveoles that are only containing minimal contamination, so to say, and significantly reduce the amount of exogenous nucleic acids, as you can also see here by the lower graph, where we are comparing the QIAGEN UCP columns compared to columns from other providers. I was already mentioning that RNA quality control is an integral part of every RNA isolation that you perform in the lab. And for liquid biopsy samples, it's especially challenging because the amounts are low and you have a lot of small RNAs within your samples. The main RNA quality parameters are listed here. Usually people are looking into quantity, so how much RNA do I have in my tube and at what concentration is it present, and do I have enough material for my downstream assay are the key questions here. Another important quality parameter is the purity. So what is really in my tube? Do I have any contamination that could be interfering with my downstream assay? And the third main parameter is the integrity. So does my RNA sample have the correct size distribution that I would expect, or is there a lot of degradation which might limit the types of analysis that I can do with my RNA sample in the downstream assay? Usually RNA quantity is assessed through the absorbance at 260 nanometers in the classical spectrophotometer. However, for this classical measurement, you need to have a certain amount of RNA in your elliot. If you have very lowly concentrated RNA samples, we do not recommend to go with the absorbance at 260 nanometers because it's not a reliable method. Instead, we are usually recommending quantitative RT-PCR. Other methods that are also used usually, like the qubit method, for example, also were not reliable in our hands, especially for small RNAs like microRNAs. So usually for liquid biopsy samples, we are recommending qRT-PCR as the method of choice to assess the RNA quantity. RNA quality and purity is usually determined by the ratio of 260 nanometers and 280 nanometers. So this ratio tells you how much contamination you have in your sample and especially how much protein you have. Another measurement is the ratio between 260 to 230, which tells you how many impurities like salts you have in your sample. Again, for liquid biopsy, this is probably not the most reliable method because you usually have very low amounts of sample here. So what you can do instead, if you want to understand how much contamination might I have in my sample, is to increase the volume of your sample and look at whether you get also more yield with increased sample volumes. If you do not see a linear increase in your yield, this might be indicating that you have some contamination in your sample. What is also important to remember is that phenol can have an effect on the UV absorbance. So this is probably then not so relevant for liquid biopsy, but it is important for other RNA isolations that you are performing. In these graphs here, you see the absorbance spectrum of phenol, and you see that the phenol spectrum overlaps with the spectrum that you would expect from pure RNA. So if you are using phenol in your workflow, it's very important to do careful pipetting of the aqueous phase and avoid carryover of phenol, because this can falsify the RNA content and make you think that you have a higher RNA content in your sample than what's actually present. RNA integrity is usually assessed through the RIN value, either through a bioanalyzer or through the QIAxcel instrument from QIAGEN. However, also for limited samples like cell-free RNA, this measurement is usually not very trustworthy. So what we are here during our product development using a lot is looking at the delta CT value between a random priming mix and an oligo-DT priming mix, as you see indicated down here. And with looking at the delta CT value, you can make an estimation on how intact your RNA is. So the larger the delta CT between the random primer mix and the oligo-DT primer mix, the less intact your RNA usually is. I also want to talk a little bit more about exosome isolation, because when you look into the exosome isolation techniques that are being used today, and this is a graphic coming from a recent publication, there is a lot of different methods that people are using today. And what I want to highlight here is that all of these methods, in fact, have their advantages and disadvantages. And what I see these days is that a lot of people start to combine different methods because just using one method is not sufficient to isolate exosomes in the desired quantity and quality. We at QIAGEN have the ExoEasy kits, and we are actually here using the technology that comes from the colleagues of Exosome Diagnostics that uses an affinity-based membrane to purify exosomes. The advantage of these kits is that they are very specific in the purification of exosomes and extracellular vesicles. They efficiently remove protein contaminants, which is some of the issue that other methods are having. And you do not need to buy any special or very expensive equipment like an ultracentrifuge. So these kits are based on a spin column, and they require a normal centrifuge that is usually present or sitting in the lab. And if we are looking a little bit more into what kind of exosomal RNAs we are detecting and how specific that enrichment is, we see that in the allele that comes from the spin column, we get an enrichment of vesicle microRNAs, so RNAs or microRNAs that are associated with vesicles, and that we get a depletion of those microRNAs that are more known to be outside of exosomes. We also get a very good capture of mRNA targets that are listed here on the right side. This is just a handful of targets that we looked at here, but you really see that we are capturing all of these targets. All of these results are also published in a publication from 2015 that we did together with our colleagues from Exosome DX, and this publication is among the top 25 most cited articles in PLOS ONE. This article also then goes a little bit more into comparing the performance of this exosomal RNA isolation technique with ultracentrifugation, and you see here that these two techniques show very comparable size distributions of the RNA that's been isolated. If you want to get a little bit more information about this technique and how it can be used also then in a downstream assay like NGS, we have recently presented a poster at the ISAF meeting this year in Canada, where we are doing exosomal microRNA profiling of colorectal cancer patients using the QIAGEN ExoRNase kit for isolation, and then the QIAGEN QIAseq microRNA library kit for small RNA library preparation. And I'm happy to send you a copy of this one if that's interesting for you. So let me sum up and slowly move over to the questions. So what I have been talking about today is mainly what matters for the sample stabilization, disruption, and then RNA isolation and QC. However, QIAGEN offers really a complete portfolio of liquid biopsy solutions, also including a lot of analytical assays that are mentioned here in the middle. As you can see, we have a broad portfolio of NGS solutions for cell-free DNA, exosomal RNA, and cell-free microRNA, and then also a complete portfolio of analysis for circulating tumor cells. And recently we've added a portfolio for circulating cell-free DNA and RNA. So this workflow would allow you to co-isolate DNA and RNA and then go subsequently into next generation sequencing as an assay. We also have a complete suite of analysis and interpretation software tools that are then supporting you in your bioinformatics analysis afterwards. So with that, I'm at the end of this presentation. I thank you very much for your attention, and you have our contact details here if you have any follow-up questions or feedback. And with that, I'm looking forward to your questions. Thank you.
Dr. Schramm, thank you so much for your informative presentation and your important research. We will now start the live portion of the Q&A in our webinar. If you have any questions you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of your presentation window. Type that question into the box that appears on the screen and we'll answer it. So let's get started. Our first question from our audience member, Dr. Schramm, is How much is DNA contamination a problem for RNA analysis from liquid biopsies?
Okay, thank you. Yeah, this is a really good question. So DNA contamination can come up in your NGS analysis, especially if you do a whole transcriptome analysis. This can be sorted out bioinformatically, so you can remove the, let's say, background that comes from DNA contamination. But of course, you're reducing the number of meaningful reads that you have in your sample or in your NGS experiment. So that's why if people want to be really on the safe side, we do recommend to do an on-column DNA treatment, and we usually give a guideline in our handbooks on how to remove DNA if that is of concern for our customers.
Thank you so much, Dr. Schramm. We have one more audience question today, and that question is, which assays do you recommend to analysis for cell-free RNA?
Yeah, this is another really good question. So for cell-free RNA, we usually recommend to go with next-generation sequencing assays. There is a lot of different options for assays available today. You can either go into a more targeted RNA sequencing approach, but you can also do a whole transcriptomics profiling of your RNA. Another option is to do specific microRNA profiling that I was also mentioning in the poster that I showed you. So preparing a specific microRNA library and then doing a, let's say, complete microRNA profiling of your samples is also possible with NGS. And for me, that would be the method of choice today. It's now affordable and maximizes the research questions that you can answer with your data set.
Dr. Schramm, thank you so much for your information. Do you have any closing remarks you'd like to share with our audience today?
Yeah, so I can tell you that personally I'm extremely thrilled and excited about all these options that are now coming up with liquid biopsy. So I'm really curious to see how this field is going to develop in the future, and I will definitely stay tuned on what new publications are coming up and where the field is developing. Thank you.
And thank you so much, Dr. Schramm. And I'd like to take the opportunity to thank LabRoots and our sponsor, QIAGEN, for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through January 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us. We hope to see you again. Bye-bye.